Hey everyone, welcome to this explainer. Today, we are diving straight into the absolute, literal building blocks of life. We're going to be exploring amino acids. Specifically, we're looking at their structure, their functions, and the really fascinating chemical properties that allow them to drive the human body. We'll see exactly how their microscopic chemistry translates into massive, life-sustaining actions. So, if you've ever wondered how a collection of simple molecules can orchestrate everything from your daily metabolism to the actual physical structure of your bones, well, you are definitely in the right place. Let's get started.
Take a look at this brilliant quote straight from our source material. It instantly sets the stakes for our journey today. Virtually every single life process you can think of absolutely depends on this specific class of macromolecules. Without them, there's no movement, no defense against disease, no metabolic regulation, nothing. But to really understand proteins, we first have to understand the microscopic units that link together to build them.
So, here is our roadmap for today's explainer. We'll be covering 1. The molecules of life, 2. Anatomy of an amino acid, 3. The 4 side chain families, 4. Acid-base buffering superpowers, and finally 5. Medical and drug applications. All right, Section 1. The molecules of life.
Proteins display an absolutely incredible diversity of functions in our bodies. For example, you have structural proteins, like collagen, which forms a framework for calcium in your bones. It literally acts like the steel cables inside reinforced concrete. Then you have transport proteins, like hemoglobin and plasma albumin, that shuttle essential molecules through your bloodstream. And defense proteins, like immunoglobulins, that fight off viruses. Yet despite this massive functional diversity, all of these proteins share one beautiful common structural feature. They are all linear polymers of amino acids.
Moving right along to Section 2, Anatomy of an amino acid. To really understand how an amino acid works, we have to look at it in its natural habitat. And for us, that natural habitat is our body's physiologic pH, which sits right around 7.4. At this specific pH, the core of the amino acid is what biochemists call a zwitterion. It's a fun word, but it basically just means it has a dipolar nature. Its carboxyl group is dissociated, giving it a negative charge, while its primary amino group is protonated, giving it a positive charge. This charged dipolar core is the foundation of literally every amino acid in your body. Now, right in the center of that zwitterion is the alpha carbon. Because this central alpha carbon is attached to 4 completely different chemical groups, it is chiral, meaning it's asymmetric. And because of this asymmetry, the molecule can actually exist in 2 distinct forms that are exact mirror images of each other. Think of it like your left and right hands. They have all the same parts, but you can't perfectly superimpose them on top of each other. In chemistry, we call these optical isomers or enantiomers. Now, this is where it gets really wild. Biology exhibits a super strict preference for just one of those mirror images. The human body exclusively synthesizes and uses the L form of amino acids to build all of its proteins. The D form, on the other hand, is pretty much relegated to foreign elements, like the cell walls of bacteria, or it's sometimes utilized in certain antibiotics. It just goes to show that your body is incredibly specific about the geometric architecture it uses to build itself. Okay, let's jump to Section 3, 4 side chain families. So while almost all amino acids share that same basic chiral core we just talked about, when they're in a protein chain, those core groups are locked up tightly in peptide bonds. Therefore, it's the distinctive side chain, or R group, that acts as the amino acid's true superpower. Nature has actually described over 300 different amino acids, but there are only 20 that are commonly found as constituents of mammalian proteins, coded directly by our DNA. We can elegantly classify these 20 amino acids into 4 distinct families, based purely on the chemical properties of their side chains. Whether they're nonpolar, uncharged polar, acidic, or basic, entirely dictates how they behave, how they interact, and ultimately how they fold a whole protein into its working shape. Let's kick things off with the nonpolar family. These side chains have a very even distribution of electrons, meaning they don't gain or lose protons, and they don't participate in hydrogen or ionic bonds. They are essentially oily or lipid-like. Because of this, they experience what's known as the hydrophobic effect. In the watery, aqueous environment of our bodies, these nonpolar side chains desperately want to hide from the water. They act just like droplets of oil in a vinaigrette, coalescing together to fill up the interior core of a folded protein, which effectively gives the protein its crucial 3-dimensional shape. But wait, within this nonpolar group, we actually have a really fascinating rebel, proline. Unlike the others,
proline's side chain curves right back around and bonds with its own alpha amino nitrogen to form a rigid five-membered ring. This makes it a secondary amino group, which is often called an amino acid. So why does this matter? Well, that unique, rigid geometry contributes directly to the tough, fibrous structure of collagen we mentioned earlier, and it often interrupts structural helices in globular proteins. It's a perfect example of structure equaling function.
And this brilliantly illustrates the uncharged polar family too. Specifically, if we look at cysteine, cysteine contains a sulfhydryl group. When two cysteines get close to each other in a protein, they can oxidize and form a super tough, covalent cross-link known as a disulfide bond. These bonds act like molecular safety pins, stabilizing extracellular proteins. For instance, albumin, that vital transport protein in your blood, relies on these exact disulfide bonds to maintain its shape while it shuttles molecules all over your body.
Next up, Section 4, Acid-Base Buffering, Superpowers.
Let's transition from static anatomy to dynamic action. How do these molecules behave when the fluid environment around them changes? It turns out they possess absolutely incredible buffering superpowers. Because amino acids contain weakly acidic carboxyl groups and weakly basic amino groups, they can act as buffers, meaning they resist wild changes in pH. If we follow the titration journey of a simple amino acid like alanine, you can see this right in action. In step 1, at a very low, highly acidic pH, alanine is fully protonated and holds a positive charge. As we add a base and raise the pH in step 2, it loses a proton from its carboxyl group to become that neutral zwitterion we discussed. Finally, in step 3, at a high basic pH, it loses another proton from its amino group, becoming fully deprotonated and negatively charged. Now keep your eye on that value, 5.7. That is a completely crucial metric for alanine. It's its isoelectric point, the exact pH where the molecule is electrically neutral. The sum of its positive charges perfectly equals the sum of its negative charges, leaving it with a net charge of 0. Every single amino acid has its own unique isoelectric point. Biochemists actually use these specific charge differences to separate and identify proteins in the lab to help diagnose diseases. This incredible ability of amino acids to swap protons and buffer against rapid pH changes isn't just magic, you know? It's actually mapped out mathematically by the Henderson-Hasselbalch equation. By calculating the exact relationship between the pH of a solution and the concentration of a weak acid and its conjugate base, this single equation allows us to predict exactly how a molecule is going to behave and what charge it will carry in any given fluid in the human body. Which brings us to our final section, Section 5, Medical and Drug Applications. So this perfectly highlights how these microscopic chemical properties and buffering equations translate into massive real-world impacts. You might think the Henderson-Hasselbalch equation is just a headache for a chemistry exam, but no, it is literally the ultimate decider of drug absorption in pharmacology. Drugs pass through our cell membranes way more readily if they are uncharged. For a weak acid like aspirin, the local pH of the stomach dictates how much of the drug achieves the uncharged HA form needed to actually permeate the membrane. For a weak base like morphine, the equation dictates how much reaches the uncharged B form. The pH at the site of absorption completely determines whether the medicine actually makes it into your bloodstream. Now, coming back to our amino acids themselves, let's take a look at histidine, which belongs to the basic side chain family. Histidine is incredibly special. It is the only amino acid with a side chain that can ionize right around our physiologic pH of 7.4. Because its side chain can easily flip between being positively charged or neutral in our natural bodily environment, it plays an absolutely vital buffering role when it's incorporated into functional proteins, like hemoglobin. It actively helps our blood resist dangerous swings in acidity. And this brings our whole explainer full circle. We've talked a lot about polar versus nonpolar and how structure defines function. Well, what happens if there's a microscopic mistake? Sickle cell anemia is a devastating sickling disease of the red blood cells, and it is caused by the replacement of just a single polar amino acid, glutamate, with a nonpolar amino acid valine at the 6th position in the beta subunit of hemoglobin. Swapping just one polar molecule for a nonpolar one completely alters how that massive protein folds and interacts, resulting in a severe systemic disease. That, right there, is the true power of the amino acid side chain. So, to wrap up this explainer, I want to leave you with this final thought. We've just explored the basic linear components, really just the mere anatomy of the individual blocks. If a single misplaced atom can fundamentally alter the architecture of our blood cells, what incredible complexities await us when we explore how these simple blocks fold together to create the massive, three-dimensional proteins that regulate literally every aspect of our lives. I really hope this structural roadmap leaves you deeply curious to explore the fascinating world of protein folding next. Thanks for learning with me today.